Now that we've discussed a bit about why things evolve the way they do, the next few lectures will be dedicated to the how part of the equation. That is, natural selection explains why certain traits spread throughout a population, because those traits increase reproductive success. But it does nothing to explain how those traits are passed from one generation to the next, or what material mechanism prevents negative traits from passing on. This question, and those that we'll examine in the next chapter, are ones that Charles Darwin and his contemporaries did not answer because they could not answer them. The basic biological research had not yet been conducted in their time. In the 19th century, no one knew enough about how living things worked to even begin answering these questions about evolutionary process. It's taken an additional 150 years of basic science to learn what we today know about evolution. That's why it's a terrible idea to refer to evolutionary theory as Darwinism. It ignores 150 years worth of subsequent discoveries and suggests that nothing has changed since Darwin's time. At any rate, to understand the mechanisms by which evolution functions, you have to start about as small as you can, on the microscopic level, because that's where all the mechanisms of evolution operate. I hope everyone at least had an introduction to biology in high school, and much of Chapter 3 in the textbook was familiar to you. If not, I'll try to point out the most important parts of the chapter today, and anything that remains unclear you can ask about on our discussion forums. First, a caveat, or maybe a reassurance. I am not a biologist, and this is not a biology course. What I'm interested in accomplishing during this lecture is to familiarize you with the biochemistry necessary to understand evolution for an Intro to Physical Anthropology class. Things will not be nearly as detailed or precise as they would be in an analogous biology course. I will simplify and gloss over things in a way that, while still accurate, might shock a biology professor. If you studied cellular biology in detail before, don't get distracted by everything I leave out. Focus instead on the points that I include and how they connect to one another to help make sense out of evolution. So to begin, we need to understand what a cell is. All life evolves and all life is made up of cells, so all the processes of evolution have to apply to cells as much as to whole organisms. In fact, since some organisms are only a single cell, all the processes of evolution have to apply completely to a single cell. But what is a cell? As your textbook puts it, cells are the fundamental units of life in all organisms. Life, as a concept, does not exist without cells. Now, there are unfortunately some fuzzy boundaries like viruses and prions that sort of act like they're alive but don't have cells. We'll just ignore them for now. But think of cells as the Legos and yourself as the little house that's built out of the Legos. By stacking up different shapes, sizes, and colors of Legos, you can build just about anything. And by putting together different kinds of cells, you can build any living organism. But what is half a Lego? It's a useless piece of weirdly shaped plastic. And half a cell isn't alive. So if we're really just a huge colony of maybe 1 trillion cooperating cells, and we evolve, the real processes of evolution must act on cells. Again, all life evolves, all life consists of cells, therefore cells must evolve. To understand how, you have to cut open the cell and look at the non-living stuff inside. A cell is kind of like a Ziploc bag full of jelly. The bag itself is called the cell membrane, and the jelly is called cytoplasm. Inside the jelly are a variety of small little structures called organelles, and it's these that carry out the necessary functions of life. It is the combination of the biochemical functions of each organelle that combines with one another to create the phenomenon we call life. 3 kinds of organelles are particularly important for us: the nucleus, the mitochondria, and the ribosomes. The nucleus is analogous to the brain of the cell. It contains the parts that ultimately direct and control the activities of the rest of the cell. The mitochondria are the power plants, breaking down nutrients and synthesizing the chemicals that the cell uses for energy. And the ribosomes are the factories, constructing the materials that the cell needs to grow and operate. All of these structures are constructed of only a few kinds of organic molecules, and ultimately it's the chemical properties of those molecules that make life and evolution possible. The most common kind of molecule in a cell is a protein. Proteins are like plastic. They can be made into almost any shape imaginable and used for almost any purpose you want. They can do this because they're really just long chains of smaller molecules called amino acids. There are 20 amino acids, and depending on what order they're linked together, the protein chain will twist and kink into the appropriate shape. Once the protein is formed, it holds that shape, just like plastic holds its shape after it cools and solidifies. This is much like how, starting with a basic limited set of beginning shapes, I can combine those shapes into any numeral 0 through 9, and I can then combine those numerals into any number at all. All I need to know is which shapes to put together in which order to create the number I want. Likewise, almost any function the cell needs to perform, the ribosomes can manufacture a protein of the appropriate shape for that purpose. But somewhere you have to have the information on which amino acids are put together in which order to feed to the ribosomes. That information is stored primarily in the cell nucleus, and the memory banks that actually record the information are therefore called nucleic acids. There are two primary forms of nucleic acid: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. There are also a couple different forms of RNA, but DNA is really more important for understanding evolution. DNA is a molecule that consists of two long chains of phosphates and sugars linked together by bases. Each smaller unit of sugar phosphate base is known as a nucleotide. If it were flattened out, DNA would look like a ladder, with the phosphate sugar chains being the uprights and each rung consisting of two bases. In reality, the ladder twists into a spiral called a double helix. It is the bases that actually record the sequence of amino acids necessary to create a protein of a certain shape. There are 4 bases in nuclear DNA: adenine, thymine, guanine, and cytosine. And because those names are so difficult, we usually just use the initial ATGC. Each nucleotide contains one base, which attaches itself to another base on the other phosphate sugar chain to create the ladder rung. But A will only bond with T and vice versa, and G will only bond with C and vice versa. And it's that property that allows DNA to record information and reproduce. Remember, anything smaller than a cell is not strictly alive, but DNA has a property very reminiscent of life in that it can replicate itself or reproduce itself. So how can a non-living molecule replicate? Well, those bonds between A and T and between C and G are very weak. At certain times in a cell's life, they break apart altogether. It's like going down a wooden ladder and sawing each rung in half. Then, rather than one 2-stranded DNA molecule, you have 2 1-stranded versions. Now, and this is the important part, these 1-stranded chains are not the only nucleic acids in the cell nucleus. There are also a lot of free-floating nucleotides. Other molecules grab those free-floating nucleotides and line them up alongside the 1-stranded DNA molecules. If an A lines up next to a T, or a C next to a G, they form a bond. If 2 nucleotides bond to the chain next to one another, their phosphate and sugar parts bind together to start forming a new chain. Eventually, each of the 1-stranded chains has formed a whole new strand. But since A's only bond with T's and C's only bond with G's, each of the new 2-stranded molecules is identical to the original. We start with a single AT pair that splits. The A finds a new T, and the T finds a new A, so we end up with 2 AT pairs. Duplicate that process a few billion times, and you have 2 completely identical DNA molecules, all just by relying on the natural chemical processes of the nuclear bases. That's how DNA replicates, but how does it encode information for making proteins? This is the process of protein synthesis, and it relies on the same basic principles. First, we must recognize that DNA's long, unbroken chain of nucleotides can be broken down conceptually into triplets, sequences of 3 nucleotides at a time. In the process of protein synthesis, each triplet will ultimately be translated into one and only one of the 20 amino acids. A protein is only a long chain of amino acids, so the sequence of DNA triplets defines a sequence of amino acids and thus a protein. Basically, sometimes the DNA chains break apart, but not entirely. When this happens, rather than forming a new DNA strand, the helper molecules assemble a complementary strand of RNA. RNA is similar to DNA, but it only ever has one strand. It also uses uracil rather than thymine, but the U just replaces the T and it still only bonds with A. The first step of protein synthesis is called transcription. A particular form of RNA, called messenger RNA or mRNA, forms on only one side of the DNA, then breaks off. The DNA triplets have governed the order of mRNA nucleotides to form a sequence of codons. Codons must be complementary to the DNA triplet they formed on, so they also specify a particular amino acid. The information in the DNA has been transcribed, that is, it's rewritten, in mRNA. When the mRNA is fully transcribed, it breaks off from its DNA, and the 2 DNA strands then close back up. The M in mRNA stands for messenger, because the mRNA then migrates to the ribosomes, carrying the message of how to construct the protein. We're now in the second phase of protein synthesis called translation. We're going to translate the information from the language of nucleic acids into the language of amino acids. The ribosome, which is mostly made up of proteins and other RNA, connects to the mRNA and begins lining up and bonding tRNA, or transfer RNA, that has been floating in the cell cytoplasm. tRNA is called transfer because its job is to transfer amino acids from the cytoplasm to the newly assembled protein. Here's the trick. At one end of each tRNA chain is a specific amino acid. At the other end is a sequence of 3 bases called an anticodon. A tRNA with a given anticodon connects only to a particular amino acid. Once you know the anticodon at one end of the molecule, you know the amino acid at the other. There's only one possibility. Because each anticodon is complementary to only one codon, the tRNA can only connect to the mRNA at a particular location. Multiple tRNA molecules must line up in a particular order, defined by the sequence of the mRNA codons. Those codons on the mRNA only occur when their complementary triplets were present in the DNA molecule. This means that the ribosome is now lining up amino acids in an order that was ultimately determined by the sequence of base pairs in the DNA. Once the amino acids are lined up, they bond to one another and release from the tRNA, and the protein chain rolls up into its proper shape. It then floats out into the cell ready to do whatever it was designed to do. So, to summarize this whole complex process, first, the order of DNA triplets governs the order of mRNA codons. Next, the order of mRNA codons governs the order that tRNA molecules align on the ribosome. The order that tRNA molecules align on the ribosome determines the order of the amino acids. Next, as we discussed before, the order of amino acids determines the shape of the protein. And finally, of course, the shape of the protein determines its function. Each step takes an input and uses it to determine an output. The output of one step becomes the input of the next step. The end result is information in the DNA turned into a functional protein molecule. DNA thus encodes recipes for synthesizing various proteins by having various sequences of base pairs. Different DNA triplets encode for different amino acids, and different triplet sequences encode for different proteins. And because of the chemical properties of the molecules, the process goes off without a hitch the vast majority of the time. But, and this is another extremely important point, it isn't 100% effective. Sometimes, very rarely, the wrong amino acids will line up, forming a bad protein. That's usually not a catastrophe because the next ribosome will produce a good version and the cell will continue unaffected. But what happens when every ribosome produces a bad version? What if, when DNA is replicating, the wrong bases line up there, forming a bad triplet? Then every protein synthesized from those triplets would have the wrong sequence of amino acids. This is called a mutation, a change in DNA sequences due to bad replication. And it's actually a very good thing. If it weren't for mutations, evolution would be impossible. More on that next chapter. But for now, let's go back to the machine when it's working properly. A gene is more or less the sequence of DNA triplets that describes a functional protein. A gene is not really a thing then, it's just part of a thing, an abstract label we use to describe a part of the DNA molecule that does something. You might think of the gene less as the molecule itself and more as the information encoded in that molecule. That is, it's not the hard drive, but the file we've stored on the hard drive. There's more to it than that, of course, and your textbook goes into some detail on how and why genes aren't as simple as we'd like them to be, but this definition will work well enough for our class. So when we talk about genetic differences between individuals or species, what we really mean is that their cells have different proteins, and they have different proteins because the sequences of DNA base pairs in their cell nuclei are different. Okay, so we know how DNA works within a cell. We know that all organisms are basically made up of huge colonies of cells working together. Crucially, with a few exceptions, every cell in an organism has identical DNA. This is because every cell in an organism derives ultimately from a single cell that divided, replicating itself identically, and replicating its DNA in the manner I just described. If a cell is primarily made of proteins, and a cell's proteins are defined by its DNA, and every cell in your body has the same DNA, then why are some cells skin cells and others are muscles or nerves? Well, not every cell is using every gene all the time. Regulatory genes influence the activity of other genes, often by creating proteins that turn other genes on or off by opening up the DNA strands. DNA can only work to make proteins when its strands have been pulled apart, like the two sides of a zipper. Regulatory genes code for proteins that work like the zipper pull. In a skin cell, the regulatory genes make proteins that turn on skin cell genes and make skin cell proteins. In muscle cells, the regulatory genes turn on the muscle cell proteins and they turn off the skin cell proteins. So that explains why cells are different, but we still haven't really looked at why they're the same. For that, we need to understand cell division. In the nucleus of most human cells, there are 46 individual DNA molecules. 23 are inherited from your father, and 23 are inherited from your mother. At certain points in the cell's life cycle, these 46 molecules bunch up and roll up into dense structures called chromosomes. Chromosomes are like genes. They're not things so much as labels for a period of time in the process of DNA replication. And that's what's going on when a DNA strand forms a chromosome. It's replicating. Chromosomes under a microscope look like X's, two strands joined at some point in the middle called a centromere. So a cell begins with 46 DNA chains. Then it begins division, so the DNA forms chromosomes and replicates. Now the cell has 46 X-shaped chromosomes, each with 2 identical DNA chains joined at the centromere. In the more common process of cell division called mitosis, the centromeres then break apart. Now there are 92 one-chain chromosomes in the cell. One DNA chain from each chromosome migrates to opposite ends of the cell, which then pinches off into 2 discrete cells. Each new cell, or daughter cell, gets one chain of DNA from each chromosome, so each cell gets 46 chromosomes identical to its parent cell. The chromosomes then unravel back into simple DNA chains. That's mitosis, which creates new somatic cells, the kind of cells throughout most of the body. But recall that I said half of the chromosomes were inherited from the father and half from the mother. That's because some cells, called gametes, have only 23 chromosomes, and they function in sexual reproduction. When one gamete from each parent comes together, they fuse their nuclei, combining their DNA into one pot, so to speak, which is a process called fertilization. This forms a zygote, the cell produced by fertilization. That cell has DNA different from each of its parent gamete cells, and it grows through mitosis into an individual with DNA different from each of its parents. That's an extremely important point too. Sexual reproduction makes evolution much easier because genes are mixed up and reshuffled every generation. More on that next chapter. But how do gametes get only half as much DNA as somatic cells? Well, that's the result of meiosis, cell division resulting in gametes. The textbook does a good job of describing the differences between mitosis and meiosis, but I'll just point out the basic difference here. In mitosis, the DNA replicates once and the cell divides once. In meiosis, the DNA replicates once, but the cell divides twice. The parent cell goes from 46 one-chain chromosomes to 46 two-chain chromosomes to 2 cells with 23 two-chain chromosomes to 4 cells with 23 one-chain chromosomes. Those 4 daughter cells are called gametes. All right then, so what have we talked about today? We started with the basic structure of the cell, nuclei, ribosomes, mitochondria. Then we looked at how DNA replicates itself and how DNA and RNA work together to create the proteins necessary for the cell to survive. And finally, how cells divide while making sure that the necessary DNA is always present. I'll leave you with one final thought to consider as you're doing the readings for the next lecture. Why does all this matter for understanding evolution and natural selection? Darwin was talking about variation within a species and which individuals had greater or fewer numbers of offspring. How does microscopic DNA affect that? Well, remember that DNA codes for proteins, and proteins are the workhorses of the cell. Every individual has unique DNA, so every individual cell has a unique complement of proteins. Sometimes 2 slightly different proteins can do the same job. Now think about the writing utensil that you're using to take notes right now. Some people in the class, no doubt, are taking notes with pencils. Others are using pens. Others might be typing in another window. All of those ways of taking notes accomplish the same function. Some, though, are better at it. This pen doesn't skip as much, or the pencil lead doesn't break as much. So the people with the best writing utensils become the best note takers. Those people are able to study better and will get better, higher grades on the exam. That means they'll get better final grades and graduate with a higher GPA. A higher GPA translates to a better job, and that means a higher wage. A higher wage means buying a bigger house, and so on. Now, obviously, this is a bit of an exaggeration, but things work much the same way in the living world. There are many kinds of proteins that get the job done, but some are better than others. Those individuals with the best proteins have cells that work the best. they become the best, most well-adapted individuals. They survive the longest. They reproduce the most. They pass on their genes to their offspring the most. And they make sure those offspring have the best adapted proteins. Very small changes at the beginning of a long series of cause and effect can produce significant changes at the end. Of course, it's not quite that simple, but we'll cover the intricacies of inheritance next chapter.